All right, in this video, I'll talk about splicing. First, I'll go over the molecular basis of what splicing actually does and is, and then I'll go over the experimental history of Drosophila sex determination, specifically with sex lethal and determination of male versus female, just the first step of the process. So let's begin with the molecular basis of splicing. Before we get started, splicing, remember, is just you have your pre-mRNA that's freshly transcribed, right? And we'll have regions that we call introns and exons. So let's say this is exon 1, this is exon 2, let's say this is exon 3. And then in the middle, let's say we have intron 1, intron 2, intron 3. And what splicing basically is that we can splice out the introns. And we can do this in a multitude of ways, right? Like, for example, we could have this splicing, and this would produce 3 exons, E1, E2, E3, right? But we can also have this splicing, right, where it goes above, and in this case, we will lose exon 2. This is just a way for us to get multiple different proteins. This is very important in, let's say, differentiation, right? Like we'll go over with sex determination, right? This can alter protein structure, protein function, a lot of cool regulatory effects that are implicative from this model. And this is something seen in eukaryotes specifically. And the way it basically works, obviously, the overall view is pretty simple, but when we actually get into the chemistry of it, it gets a little complicated. First, some quick things. This is an exon. This is another exon. And in here, this is all an intron. And what we see is we have something called the branch point, and branch point A is what it's usually called. And then obviously adenosine, it has an OH group on top, right? And basically what this adenosine hydroxyl group will do is that, remember there's a few electrons on here, right, from organic chemistry. This will nucleophilically attack the phosphodiester bond right there. And it'll basically connect itself. If you see after step 1, it'll connect the A. Here, I'll highlight it actually. Oops. We see the A, right? And then we see the P, and then we see the G. What color should I do? I'll do yellow. GU. And we can basically see is that it ends up, forming like a little loop with itself, right? And then this phosphodiester bond over here, with this G, it breaks off, as you can see here, and you're left with just a hydroxyl group, right? And then what happens is that this hydroxyl group now, right, this one, will now nucleophilically attack the other phosphodiester bond on the other side. And then what you end up happening is the exact same thing, right? Same thing up here. And you see that this G ends up here, right? If you see this G ends up there. And then you get a phosphodiester bond between the two exons. And so this G doesn't end up there. Sorry. This G ends up here. This G is actually just chilling there still. It's just this will form a bond here. Or I guess more so, it breaks the bond and forms a new one with this exon, right? It'll break this phosphodiester bond between these two, and then it'll form a phosphodiester bond with this, let's say it's like a C or something, right? And then what happens is you get an excision of a lariat structure, and this guy kind of just goes away, right? And then what we end up with are two exons, exon, and that's basically the mechanism of splicing. Obviously, this is not just going to happen spontaneously. Usually we need like helper molecules, and that's basically what we see here. There's a lot is on here, right? But right now, I just want to talk about one big one, which is this U2AF protein here. And actually, before I point that out, notice that we have 3 different parts of this exon-intron-exon junction, right? We have the 5' splice site, right? Remember, this is like this side of the junction. We have the branch point. Remember the branch point A. We see an A is there, right? Remember, this hydroxyl group will come and attack this guy, right? And then this guy will attack this guy, and we break off the lariat, right? And then we also have the 3' splice site, which is just over here. And so what I want you to bring attention to is this 3' splice site. We have 2 proteins bound to it, but we just want to focus on U2AF. Okay, U2AF is an important one. Let's keep that in mind. U2AF binds to the 3' splice site. That's about it. I want you to know for this slide, because now I'm going to get into Drosophila sex determination. And I'll just give a quick overview of Drosophila sex determination. Let's begin our story with a developing embryo. So we have a female embryo and a male embryo. Obviously, since they're female and male, there will be some differences. And the biggest difference is that in the male embryo, originally, this is the sex lethal gene right here, and there is no transcription. It doesn't work. Whereas in the female one, there is this PE promoter, all right? And this PE promoter will just transcribe functional sex lethal protein, right? This is what they call early female sex lethal protein. And what this guy can basically do is that later in embryogenesis, this promoter stops functioning, okay? So the promoter no longer functions properly. So now we use this PL promoter. I guess L can stand for later in embryogenesis, whereas E stands for early. And then what you end up getting is this protein, right? Ignore this guy for now. You get this protein, which is the same thing that you get in the male. All right, the PL protein also, the promoter also starts working, and so you get this protein. Now the difference, right? Let's see what the difference is. In the female, there is expression of this sex lethal protein right here, right? In the male, there's nothing. There's no sex lethal protein, and so what you end up seeing is that this sex lethal protein is actually a splicing repressor. And it'll actually repress splicing at this exon 3, right? This is exon 3. And what you get is this series of exons in your process mRNA. And this encodes for late sex lethal protein, which is functional, okay? I remember, it's functional. Whereas when we look at the male, right, if we look at the male, there is no sex lethal protein, so there is no repression of splicing at exon 3, and so you just get your normal 1, 2, 3, 4, right? Exon 1, 2, 3, 4, and this actually encodes for a non-functional sex lethal protein. This is a very good example of different examples of you don't necessarily need splicing for functional protein. In fact, in this case, you need less splicing for a functional protein, right? And so now we have a functional sex lethal protein in females that is, being expressed pretty often. Remember, this up here was transient, right? This was a transient expression of sex lethal protein, but you only need a little bit because this is a positive feedback loop, right? As you get more of this sex lethal protein encoded, right, more of it's going to go and bind back onto the female pre-mRNA, and then you'll get more functional ones. Whereas the male, they kind of got nothing going on for them, right? They kind of just exons 1 through 4. And so what you see with this sex lethal protein, right? So that's basically what we covered up here, right? What this functional protein can do is it actually blocks splicing again, but at a different gene. And this is what we call the transformer gene, or the TRA gene. And this encodes for a TRA protein, right? And that kind of makes sense. And pretty much exact same function, except it's at exon 2 instead of exon 2 instead of exon 3. And so in females, you get just exons 1 and 3, right? And then in males, you get both 1, 2, and 3, because remember, we had non-functional sex lethal protein in males. And so now you have, these are the differences. And actually, exact same as sex lethal, the female one is functional. All right, the male one, this guy, he's poop, right? And he can't really do anything. But what this functional TRA protein does, all right, is it is a splicing activator at the double sex gene. So there's another gene, I remember, so there's 3 genes: sex lethal, transformer, and double sex. And double sex, normally you have splicing between 3 and 5, okay? That's like what the male will end up having, because there's no TRA protein, right? So splicing is not affected. Now in in females, your TRA protein will actually activate splicing. So instead of you get 3 to 5, you just get 3 to 4, right? And this 5 is lost, Okay, so you end up just getting exons 3 and 4 in your mRNA after processing, right? So you see this difference. Female has 3 and 4, male has 3 and 5. This was because the female had functional TRA protein, which came from functional sex lethal protein, whereas the male, their splicing is not effective whatsoever, so it's just a default, right? The default is 3 to 5. This is because the TRA protein wasn't functional because there was no sex lethal protein. So you just got 1, 2, 3. And this was ultimately because in females there was transient expression of sex lethal protein, whereas in males there was nothing, right? They kind of got, they got nothing. And what these double sex protein does, right, is that they go and regulate sex differentiation in the genome. Okay, so the female double sex protein, right, will go and repress male differentiation genes, and likewise the male double sex protein will go and suppress female differentiation genes, and this is basically how sex is determined in Drosophila. That was quite a bit, but hopefully it makes sense. But now I want to go over how they even figure this out in the first place, right? This is kind of wonky, right? Well, they obviously had these hypotheses, and so they wanted to test them in like a lab setting. And first, they wanted to see if sex lethal regulates transformer. Remember, transformer was what was this gene here, right? And remember, in our model, double sex goes in females, double sex goes and represses splicing. And so we're sort of trying to see if there's evidence for this model, right? And so what they did is that they had a nuclear extract from male cells, right? So male cells, they should have non-functional sex lethal proteins, right? And then they added TRA pre-mRNA, right? So basically what you get is this, right? And remember, we're in males, so we should not have any functional sex lethal protein. This is just this mRNA minus this functional protein. So in a normal case, right, this would end up just splicing like this normally, right? So you get 1, 2, 3, right? That's basically what we're thinking. But then they also added GST tagged sex lethal, functional sex lethal, I should say, functional, right? And they started off without any, and then they started increasing the concentration, as you can see on the right. And what they saw, if you see, as you add more functional sex lethal, what you see is that the male splice starts fading away, and then the female splice starts appearing. Okay, and obviously the female splice is lower on because, or smaller because, well, that kind of tracks, right? If we're splicing between 1 and 3 now, you're gonna lose this second exon, meaning your mRNA is gonna be smaller, right? And so this supports the model. Another interesting thing is that you can see that okay, this pre-mRNA, right, that has all of them, has everything, right, actually starts increasing as you add sex lethal, which suggests that either this experiment wasn't super perfect in making these splices, or that splicing itself is not that efficient when you have sex lethal protein in the first place. So that's very interesting. But the big takeaway: male splice decreased, female splice increased when you added functional sex lethal. Right? That supports our model. This is good. And then later on, other work showed that this binding is at the 3' splice site. Remember, if we take a look here, 3', right? 3' splice site. Remember, there's a 5', there's the branch point A, and there's the 3', right? And so some work showed that this was at the 3' splice site. But if I direct you all the way to the beginning, what also binds at the 3' splice site? See that? U2AF. He comes back. We're back with U2AF. All right. So U2AF also binds there. And so is there like some sort of some beef between these two? Right. We're trying to figure that out. That's sort of what the next step was, just to see, how does it work even more in detail. How does U2AF play in all this? So they ran a gel shift assay, right? And basically what this is you in vitro, right? You add in your probe. Your probe is just, in this case, would be your like the pre-mRNA, right? And then you add in your protein. So in this case, they would add sex lethal, right? And what they should see is that sex lethal will bind on, and this protein complex will be bigger and it'll run slower on a gel, right? So you should be able to see a difference in band migration length, right? And obviously in this experiment, they also had like U2AF, right? They also have U2AF in the solution because it's from a cell extract, right? And so they saw if you just look at in this, they didn't have U2AF in the solution. They just had sex lethal, right? I can't claim this. Hold on. I cannot claim this. Binding affinity. Because there's no U2AF. So we know that sex lethal has affinity to the 3' splice site. Actually, the male 3' splice site specifically, right? Which should make sense, right? Because if we're looking over here, right, the male splice site, right, is here, and the female splice site is over here, right? And if we're thinking about it, sex lethal should only be able to bind to this splice site, right? Even though sex lethal doesn't bind to the male splice site, right? Because in males, sex lethal is non-functional, but it is this specific splice site that is important. It might be easier to think of it as a 3' splice site of exon 3, right? Right here. Whereas it wouldn't bind over here, right? This is what a female 3' splice site would be. So this should make sense, right? So, okay, there is DNA binding affinity here, but they want to see, okay, does U2AF also bind here? And like, how does that work exactly, right? And so what they did was they looked at the KD values. So the KD value is basically the concentration you need, where you get, you see this like half and half, right? When you get half of the probe shifted up, half of it is not shifted up, right? And what they found was that the male 3' splice site, right? Remember, this is exon 2. Or maybe I should have this diagram here, right? This is the male splice, this is the female splice, right? This is exon 1, exon 2, exon 3. The male splice site is right here, right? Okay, and let's take a look at this. You only need 10 to the negative 9 molar of sex lethal to get half of the shift, right? But U2AF, okay, well, you need less. Oh, sorry, you need more. You need a higher concentration, right? By like a factor of 10. So that's actually quite a big difference. And so this suggests that, okay, sex lethal can bind better to here. But here's the key. Here's the kicker. You see that the female splice site, there's no binding of sex lethal, which should make sense, right? Because the sex lethal represses splicing here, not here, right? It represses it here, right? But you see, look at how high you need of a okay, but so then if we look at the female splice site, right, this sort of makes sense. There's no binding at all with sex lethal, right? Because that kind of makes sense because sex lethal is meant to repress splicing here, right, between E1 E2, not here, right? Because we see in females there's splicing over here, right? So it makes sense that sex lethal can't bind here. And obviously U2AF, it can bind there, right? Which also makes sense if it's involved in splicing, right? I mean, it kind of has to be there, right? So this makes sense. This is very consistent with our model so far. But we want to investigate this a bit more, okay? Does this mean that sex lethal and U2AF both have binding affinity to this splice site? But is there like competitive inhibition? Like what is this, right? Like what is this mechanism exactly, right? How does sex lethal repress splicing? So we're sort of like thinking, okay, it's probably competitive inhibition, right? Because they both have affinity. Sex lethal has higher affinity for just this specific splice site. So maybe that's what we see, right? And so what they did was they did a UV cross-linking assay, right? And if you see here, you just have a bunch of proteins, right? So, for example, these proteins would be our sex lethal and U2AF, right? And then you have our DNA strands that are labeled, right? So, or as RNA, sorry. And RNA strands, these are just our pre-mRNAs, right? And then you irritate it with UV light, or irradiate it, not irritate it, irradiate, and you end up getting a covalent linkage that is irreversible, actually. Like, you can't reverse it, but that doesn't really matter. So you now have covalent linkage. You digest it with RNA, and then you run it on a gel, right? And what you should see is the proteins that are like isolated from it, right? The proteins that end up binding to the DNA, okay? And what they found was, if you take a look here, they plotted respective concentrations and U2AF, right? So now you can directly see how the two interact when you change their concentrations together, right? Here it was just measuring in like an in vitro experiment how much you need, right? And this just independently shows that sex lethal has a higher affinity than U2AF, but this doesn't actually show their interaction, right? These are separate, well, same experiment but separate, like, tubes, right? It doesn't actually measure like their interaction, whereas this does, because here we can see we're like customizing the respective concentrations, and we see that as soon as sex lethal is even just a third of what U2AF is, we see like a very good like balance between those two. And then as soon as sex lethal is like the same concentration, boom, all sex lethal, all of it, right? And then even if you go higher, right, you get crazy amounts of sex lethal. And so this was very, very suggestive of competitive inhibition, right? Because when you put both in the same solution, sex lethal wins. All right, so that kind of suggests that, okay, the way this repression works is that sex lethal binds there competitively at the active site, so U2AF can't get there and thus can't do splicing there. So that's sort of our final model. We see that in the female splicing, sex lethal binds here, so that U2AF can't come here, and so we'll instead go here, right? Well, usually, yeah, usually it still goes there anyway, but like, it'll go there instead, and then it'll make this splice cut that we see down here. Whereas in males, obviously our sex lethal can't bind there, right? Because it's non-functional in males. We have non-functional sex lethal protein. If it was functional, then we would get this female splice. But since we're in males, right, remember they have non-functional sex lethal protein. If we go all the way back, right, all the way back, because they had no transient expression of functional sex lethal protein to begin with, and so they never actually got a chance to make sex lethal protein, right? And so remember, we're looking at the transformer gene, right? So we're not even looking at like we're not even looking at sex lethal itself. We're looking at just transformer gene. So this is essentially the model that we've kind of like. Well, we haven't. To be fair, we haven't supported the whole thing. This is literally just like the first step, right? By the way, this is exactly how they figured out how sex lethal works as a repressor, right? And they found that it was competitively inhibiting U2AF at the 3' splice site. And obviously, since sex lethal protein is only functional in females, that's why we see this big cut with females, whereas in males, they have non-functional sex lethal protein. So only U2AF binds to this 3' splice site. And so then we get splicing earlier, and therefore we get our non-functional TRA protein. And the same function goes for sex lethal gene as well.